How's everybody doing today? Wonderful. How many moms do we have here today? One back there. A couple back over here. Did they tell you about the initiation for this class, what moms have to do in here? They didn't tell you about that? Did they tell you what they've been doing? Let me tell you what they've really been doing, okay? Now, there's a requirement in this class that there's an activity that everybody has to participate into, and I think moms should participate in this activity too. Don't you guys think so? Yes. I think so. So, I know that your sons and daughters have been telling you certain things about this class, but I want to set the record straight. So, I think we should do that right now to start, okay? Just so we're all on the same page, okay? Okay, moms, you're going to have to sing real loud. All right? It's to the tune of an old song called Danny Boy, okay? Oh, mommy dear, this is my biochemistry. The problem's long, the course is really tough. That last exam, it really put the fear in me. I studied lots, I hope it was enough. But let's forget about it while you're visiting and have some fun when we get out of class. Then when you're gone, I'll go back to my studying. That Kevin Ahern really, truly is an... I was a little worried about that ending there. I wasn't quite sure how that was going to turn out. So it can get ugly in here sometimes, so you got to be careful. So anyway, welcome. I hope you guys are having a good day and you have a great weekend. So it's fun. Okay. What I want to do today is a couple things. One, before I get started, I have scheduled a review session. We have an exam on Tuesday, and I've scheduled a review session in this room after class on Monday. Okay? So at 5 o'clock on Monday, I was going to have it on Sunday, but it turned out it was impractical to get a room like I wanted. But we'll have it in here on Monday at 5 o'clock. I will videotape it, and I will make the videotape available as I've done before. Okay? All right. So that's one thing. The second thing is the question has come up, where am I stopping? Somebody lost their battery. Oh! I lost my battery. It works. Okay, good. You're an absent-minded professor, right? Where to stop the material for the exam. I've decided to stop the material at the end of transcription. Okay? So where I finish transcription here, that'll be the material for the exam. Okay? Everybody clear on that? So now you can have a much more time over the weekend to goof off because there'll be less material they have to learn, right? Okay. All right. So last time I finished talking about some interesting considerations of eukaryotic transcription. I'm losing my voice now. Eukaryotic transcription. And as I said, the process is, the overall process is similar, but boy, the details are really different. And we didn't focus a lot on the details. So I think what I said would really, is really the level of depth I would expect you to know eukaryotic transcription. The other things I'm going to say here are partly about some general considerations of DNA binding proteins, and then I'm going to come back down and talk just a little bit more about some considerations of eukaryotes, because again, there's some unusual things that are there. All right? Well, I've been talking about transcription factors, and transcription factors, you should recall, are proteins that bind to DNA, and they usually bind to specific sequences in DNA. Okay? And that's how they help the RNA polymerase to recognize a promoter, for example. That may be how they stop transcription. It might be how they start transcription, a variety of things that they can do. What you see on the screen and the three slides I'm going to show you are schematic representations of proteins that bind to regions of DNA. Okay? And these proteins have a common structural feature to them. That is, they're different proteins, but all of them have a very similar structure. Okay? a similar structure. This similar structure that whenever we see a similar structure appearing across different proteins, we give it a name, and we call that name a motif. It's called a structural motif. So one of the structural motifs we see with respect to DNA binding proteins is something called a helix-turn helix. That's what the HTH refers to there. Okay? And we can see that you might wonder, in fact, I had a couple questions last time about, well, how is it that a protein can recognize the sequence of DNA? You can see that the way this protein orients itself is it's actually sitting right there in the middle of that major groove, and it's actually reading those bases. That is, it knows the sequence that it sees there. And so that sort of intimate interaction that the protein has with the major groove allows it to literally read the sequence of DNA. And so proteins that have this helix-turn helix will bind just as I'm showing you here. Another interesting set of proteins that bind to DNA are called zinc fingers. And zinc fingers are kind of cool, okay? They derive their name from the fact that they all have a zinc, and that zinc is at the center of a region that's bounded by 4 amino acids, cysteines and histidines that look like this. And the shape that it makes with this loop as a result of this binding, it looks like a finger. And this finger can stick itself into the DNA as well. Okay? So a zinc finger is another type of structural motif that is important. You can see they can be arranged in a variety of ways. But suffice it to say that they all have some sort of an element of a finger associated with them. And there again, you can see the interactions there. Again, in this case, interacting with the major groove, as you can see. Okay, and the last of these is called a leucine zipper. Now, leucine zippers can also interact with other proteins in addition to interacting with DNA. But leucine zippers, particularly when they're interacting with other proteins, have a very interesting feature. Okay? So what you see is this amino acid sequence of this protein. And you notice that about every 7 amino acids, you see leucine, 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 leucine. They all line up with each other. Okay? And the other amino acids don't line up with each other. But the leucine, 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 leucines all line up with each other. Now, what leucine zippers do is they make a structure that's kind of like a zipper. Okay? So the leucines, you may recall, have a hydrophobic side chain. So I might have a leucine zipper that's running along my finger here, okay? And on the outside of my finger is every 7 residues, I've got a leucine that's sticking out there, okay? Well, those leucine side chains are fairly hydrophobic, and you remember that we're in an aqueous environment in the cell, that is, it's surrounded by water, and those leucines really don't like to be sticking out in water. So what they will commonly do is they'll find another leucine zipper, and guess what? They interact like this. The side chains of the leucines are interacting with each other because they're both hydrophobic. They're both avoiding water. And now they make a structure that's like this that can now go down and interact with the DNA. Kind of cool. Okay? Leucine zippers are involved in quite a variety of things, but this is one very interesting arrangement of leucine zippers that allows either 2 proteins or 2 parts of the same protein to interact and have some structural integrity that the bottom part here would then interact with the DNA. So kind of cool. Okay, so those are the things I want to say about just general structural motifs. Yes, sir? Do zinc fingers all have cysteine and histidine? Yes, they do. Yes, they do. Okay. Well, the last things I want to talk about are modifications to RNA. So we didn't really talk too much about modifications to DNA. And yes, DNA does get modified a little bit, but it's kind of beyond the scope of this course to talk too much about that. But RNA gets modified a lot. When I'm talking about modifying, I'm talking about chemical modification that's occurring to the RNAs. Okay? tRNAs are very, very highly modified. Very, very highly modified. Meaning that there may be methyl groups, there may be hydroxyls, there may be various other things that are done to the bases inside of a tRNA to chemically modify them. And it's not completely understood why that occurs. It may be to help in the translation process. It may be to help flag, hey, this is a tRNA and not something else. And there may be other reasons why those modifications are done as well. For our purpose, the fact that they get very highly modified is worth noting. Now, that modification in tRNAs occurs in both prokaryotic and eukaryotic cells. Okay? Both prokaryotic and eukaryotic cells. Messenger RNAs, on the other hand, are very different between prokaryotic and eukaryotic cells. In prokaryotes, messenger RNAs are not hardly modified at all. In eukaryotic cells, messenger RNAs are extensively modified. Extensively modified. So a big difference between prokaryotes and eukaryotes relates to messenger RNAs. All right. Well, what kinds of modification happen? All right. Well, the first of these is what's called a cap. Okay? At the 5' end of a messenger RNA in a eukaryotic cell, there's a structure that's put onto it called a cap. And the cap looks chemically like this. No, you're not going to need to draw that on an exam. But I'll tell you what's there. It has a guanine, it's actually a modified guanine base that's attached. Here was the original 5' end right here. So that was the 5' end of this messenger RNA. We'll go 5' to 3' going down. To that 5' end is attached a modified guanine, I said base, a modified guanine nucleotide. And that modified guanine nucleotide is arranged in an unusual structure. It's the only place in biology where we see a 5' to 5' linkage. This is the 5' end of this guy. This is the 5' end of this guy. There was the original 5'. Now the 5' end of this guy is attached to it. So it's a 5' to 5' attachment. And you might wonder, why in the world does the cell do that? Okay? Well, there's a couple reasons. One is this structure is actually recognized by the translational machinery, which we'll talk about later today. And the other is that this protects the messenger RNA from being degraded by nucleases. Okay? From being degraded by nucleases. Nucleases are enzymes that break down nucleic acids. We talked about an exonuclease. You may remember that was a part of the DNA polymerase. We had a 3' to 5' exonuclease. We had a 5' to 3' exonuclease. And somebody asked me in my office the other day, are exonucleases separate from polymerases? And the answer is there are some that are, yeah. Okay? One kind of nuclease that's very, very abundant in cells is called RNase, R-N-A-S-E. And RNase loves to cut up RNAs. And you might wonder, well, why in the world does the cell have something that likes to cut up RNAs? Because shouldn't the cell be protecting its RNAs like it protects its DNAs? And the answer is no. Okay? No. Because I hope what you've been taking away from what I've been talking about messenger RNAs is that the level of those is important for any given cell. So a cell says, okay, I need to make a messenger RNA for troponin. I'm a muscle cell. And the skin cell says, I don't need to make that. That's different. But the muscle cell might need to make the troponin at some times and not at other times. So cells are what I like to describe as control freaks. If they make something, they like to be able to break it back down. And so one of the ways that a cell can control how much of a given messenger RNA it has is by how much it makes, how fast it makes it, and by how fast it breaks it down. Okay? Now, in a bacterial cell, this process happens really rapidly, making it and breaking it down. Bacteria will take a messenger RNA, and many messenger RNAs in bacteria have a half-life, meaning a half-time of degradation, of about 15 minutes. In eukaryotic cells, that half-life may be up to 24 hours, or maybe even longer in some cases. Okay? Well, one of the reasons that they're stabler in eukaryotic cells, remember, eukaryotic cells are not dividing as rapidly as prokaryotic cells, so minute-to-minute changes aren't quite so important is because these 5' caps are protecting against this happening. So the 5' end really doesn't suffer much in the way of degradation by nucleases. We'll see that happens at the 3' end primarily. Okay. A second modification that occurs to eukaryotic messenger RNAs that doesn't happen to prokaryotic messenger RNAs is the attachment of what's called a poly-A tail. Poly-A tail. As its name suggests, that means that at the 3' end of the messenger RNA, there's a long sequence of A's that gets attached. So once the RNA has gotten to the point where it's finished being made, there's an enzyme called poly-A polymerase. And what it'll do is it'll grab a hold of the end of the 3' end of the messenger RNA, and it'll start adding A, hundreds of these. You talk about a waste of time and a waste of energy. Why does the cell do that? Well, again, two reasons. One, it is involved in the translational process in eukaryotes. And second, this provides stability also. It doesn't stop nucleases from working, but it does slow them down. The longer the A's are, the longer it takes a nuclease to chew it back. And it turns out that the longer the sequence of A's is, the longer the messenger RNA hangs around the cell. And the reason is, again, because the nucleases are just taking longer to chew down that long poly-A sequence. I see some frowns. You're frowning. Does that make sense? Okay. You don't have to say yeah. Yes, question? So the first reason is, just like the cap, that the poly-A tail plays a role in translation, that is the synthesis of protein in eukaryotic cells. Remember, the messenger RNA is what's being translated to make protein. Yes, sir? The name of the enzyme that puts the A's onto the messenger RNA is known as poly-A polymerase. Yes?
So are there not nucleases that break down from the 3' end?
So there are nucleases that break down from the 3' end.
From the 5'.
But not from the 5' end, because the cap really stops that from happening. That's right.
Except 5'.
What's that?
Except for the 5', the 3'. I think it's it.
So a 5' and 3' exonuclease that I talked about is working on DNA. We're talking about RNA here. Okay? So, I see what you're talking about. I'm sorry. Back up. That's not true. 5' to 3', we're talking about in prokaryotic cells, okay, moving to remove the primer. That is removing an RNA, but remember that's a prokaryotic cell that doesn't have a cap on it.
Okay.
So this is only in eukaryotes. So even where we have a 5' to 3' exonuclease, it's not going to touch this because the cap is stopping that from happening.
And that's the only 5' to 3'?
That is the 5' to 3' exonuclease that would be relevant, yes. That's the only place you're going to see that. Good question, though. Good question. Okay. Well, the last thing I'm going to talk about, I mentioned before, is the phenomenon I mentioned in class called splicing. And splicing only occurs in eukaryotic cells with two very minor exceptions in prokaryotic cells. For the most part, we talk about it in eukaryotic cells. Splicing occurs on all different kinds of RNA. It can happen to tRNAs, it can happen to ribosomal RNAs, and it can happen to messenger RNAs. Okay? This happens in eukaryotic cells because I said that in eukaryotic cells, we don't have operons. We have everything on one piece of RNA, right? But I said those genes were in pieces. So I might have a messenger RNA that's this long. Here's a segment here, a segment here, and a segment here that end up being in the final messenger RNA. Well, how do they start out here and end up in a final? What happens is, if I've got a here, and a here, the pieces in between have to get chopped out, and the other pieces have to be joined together. That's called splicing. Okay? Splicing is a process that when it was discovered, like other processes I've talked about this term, people scratch their heads and said, "You've got to be kidding me." Why in the world would cells have something so complicated to do this? And the more they looked, the more they discovered that it was all over the eukaryotic kingdom. Everywhere. Most genes in our cells are spliced. Now, there are probably reasons relating to evolutionary diversity and rapid evolution of proteins. That's why it's there. I'm not going to talk about those today. But suffice it to say that this process, that this phenomenon is very widespread. Okay? There are some genes that may have 50 introns that get removed. This may span over 1 million bases, okay, this process. So in some cases, it's very, very complicated. There's a protein involved in muscular dystrophy called dystrophonin. And dystrophonin is one of the largest proteins known, and I think it stretches over 1 million bases. Okay? And it has to be spliced properly in order to get those exons together. And if it's not, it makes a non-functional protein. In some cases, it's thought that dystrophonin may, in fact, play a role in the improper splicing may play a role in the progression of the disease. Okay. That's the end of the material for the exam, for the second exam. Any questions before I move forward? Yes? Does splicing occur in prokaryotic cells? For the purpose of this class, we'll say no. The true answer is that there are, I am only aware of 2 genes in prokaryotic cells that are spliced, and they're sort of unusual. So the answer is basically no. Okay. Well, we turn our attention now. We're going through the central dogma. Remember, the first step was the replication of DNA. The second step was the transcription to make RNA. And the third step is the synthesis of protein. And they all work together to make the most important things in our cells, the proteins. The proteins are, as I described in the lectures earlier, the workhorses of the cell. Without proteins, we are hosed. It's why we cook our food, to kill bacteria, because we denature the proteins and the cells therefore die. It's why we wash our hands. We denature the proteins in those bacteria, and the bacteria therefore die. Okay? So proteins are pretty critical. The process of protein synthesis, you're going to see, is fairly intricate. All right? There are several steps to the process. All right? And I'm going to talk about them. In this case, we talk about actually 4 steps. 4 steps. Some people include the first one. Other people don't include it as part of that. But we're going to include it. Amino acid activation. And what this means is how it is that an amino acid gets attached to a transfer RNA. Remember, the transfer RNA is the thing that carries the amino acid to the place where protein synthesis is occurring. So amino acid activation is how the amino acid gets attached to the proper transfer RNA. The last 3 steps in the process all occur in specialized structure I'm sure you've heard about before, called the ribosome. So the ribosome is a complex of proteins and ribosomal RNAs that come together to synthesize protein. And they synthesize protein using information in the messenger RNAs. So the messenger RNAs provide the information. The ribosomes provide the factory necessary to make the protein. It's pretty cool. People compare the messenger RNAs to the blueprint necessary to make something, and there's some analogy to that. But I really like the description of the ribosome as the factory, because the ribosome is the place where the protein is actually made, just like an automobile is made in a factory as well. The 3 steps here of initiation, elongation, and termination all occur within the confines of the ribosome. The first step here does not occur in the ribosome. It occurs apart from it. Well, when we talk about translation, I said that the messenger RNA contained the information necessary to make protein. The messenger, that information in the messenger RNA is encoded. The nature of the code is called the genetic code, and the genetic code is pretty darn cool too. The genetic code is expressed in what we call an alphabet of 3 letters. Those 3 letters correspond to bases that are in the messenger RNA. And you can read the genetic code as follows. If we start at the 5' end, and now we're reading the sequence in the messenger RNA, the first base is U. If the second base is U and the third base is G, we would have a UUG, and that would correspond to the amino acid leucine. This would tell the ribosome, when it encounters this sequence, to put a leucine in here. Okay? I love leucine and she loves me, right? That's a joke. It wasn't a very good joke, but it was a joke. You like that? You want the A, don't you? You can have the A. Okay, you get the A. All right. So we have a genetic code that looks like this. All right. And as we look at the genetic code, we have 4 bases, and there are 3 of them. We end up with 4 to the 3rd power. That's 4 times 4 times 4. That means that there are 64 possible 3-base sequences. 64 possible. Okay? This corresponds to, and these each correspond, you'll see that there's what we call redundancy in the code. And the redundancy refers to the fact that some amino acids are coded for by more than one 3-letter code. If we look, for example, at CUU, we see it's leucine. If we look at CUC, we see it's leucine. If we look at CUA, it's leucine. And if we see CUG, it's leucine. We could describe leucine as being coded for CUX, where X refers to any nucleotide. Okay? Any nucleotide. That's not uncommon. Look at valine. Same thing. Look at alanine. Same thing. You might say, well, why do we have a 3-letter code if we have all this redundancy? Well, if we only had a 2-letter code, we would only have 16 possible 2-base sequences. And there are 20 amino acids. We couldn't cover everything. So we got 64. We have plenty of room to have more than one for each one of these. Now, I'm not going to expect you to know the genetic code, obviously. But there are some sequences I think that you should know. There are 4 magic sequences in the genetic code. The first of these is called AUG. Okay? So here's A. We go over to U, and we go down to G, and we see it codes for methionine. And you see a little asterisk next to methionine. AUG is what's known as the initiation codon. By the way, a 3-base sequence is a codon. Each one of these is a codon up here. All right? AUG corresponds to a codon. The initiation codon tells the ribosome, hey, this is the place to start. Because methionine is almost always the first amino acid that's put into a protein, whether it's prokaryotic or eukaryotic. Methionine is almost always the very first one that's put in. So it makes sense to call it the initiation codon. Well, the ribosome has to know where to start, but it also has to know where to stop. And to stop, there are 3 codons that the ribosome reads to make sure they stop. Stop signs are pretty important. You don't stop out in the real world and you get a ticket if it's good, or you hit somebody if it's bad, right? All right? So 3 stop codons. UA is a stop codon. That is I'm not saying this right. UAA, now I can't even say it right. UAA is a stop codon. UAG, you see, is a stop codon. Okay? And UGA is a stop codon. All 3 of those will stop the protein from being made. Okay. Now you see some interesting things here. Not all these go for 4. Here's AAU, AAC. Both of those code for asparagine. And then we have AAA, AAG. They both code for lysine. In both cases, we see this is AA pyrimidine, AA pyrimidine. This is AA purine, AA purine. Same thing down here. GA pyrimidine, GA pyrimidine. So there's some organization to this. It's not just a random code. Now, as I said, you don't need to memorize this code or anything. I think you should know those 4 special ones I told you. All right? But what's interesting about the genetic code is it really is what we call universal. Bacteria that are living in your gut, single-celled, simple organisms, are using exactly the same code that your cells are using. Virtually every cell on the face of the earth uses exactly the same code. Okay? The handful of exceptions where there's changes in the genetic code are very minor. Some organisms may have one codon that's different. And that's it. Okay? So the genetic code is what we call universal because it codes for all. I mean, the code is prevalent across all of living systems. Okay. Now, if we think about that codon, and by the way, this shows a relationship between a codon in the messenger RNA and the sequence it pairs with in the transfer RNA. Here's a transfer RNA, and you may remember when I showed you the sequence of that clover leaf-like structure for the transfer RNA, I said that there's a section called the anti-codon. The anti-codon is the part that pairs with the codon. The anti-codon is the sequence in the transfer RNA. It's a 3-base sequence that's complementary to the codon sequence in the messenger RNA. Now let's think back to this codon. I showed you in the genetic code several examples where you could have, for example, a GCX where the third nucleotide really didn't matter. They were all the same amino acid. That third base of the codon, okay, is what we call the wobble position. The wobble position. Meaning that that third base of the codon can be virtually anything in many cases without there being a problem. Well, cells take advantage of that. Cells take advantage of that. It means they don't have to make 64 different transfer RNAs. For a given amino acid, the first 2 bases are the most important one, and the third one is less important. And so if it doesn't really matter in the third base, sometimes what the transfer RNA will have is a modified base called inosine, I-N-O-S-I-N-E. Inosine is complementary to many nucleotides. It's reflecting the fact that I could use one transfer RNA to base pair with any of the GCXs, for example. So it means the cell has to make fewer transfer RNAs. It doesn't have to make as many transfer RNAs. Okay? Yeah, question. Does the wobble base lead to mutations? No. Okay? The wobble base also means that mutations don't really affect that position. Right? So it's actually the opposite. It means that it's much more tolerant of mutation than the other bases actually are. Good question. Other questions? Okay. All right. That just shows you all the things that I can pair with. You don't need to memorize that, but it just shows you that I can pair with any of those. And as a consequence, the cell has to make fewer transfer RNAs. One of the things that we see, actually, it's interesting in RNA compared to DNA, is that in DNA, GT base pairs are not stable, but in RNA, GU base pairs actually are. So G can pair with either C or U, as you see here. And again, I'm just showing you the information, so don't get too hung up on that. All right. Let's talk about that first step in the process, that amino acid activation that occurs. Okay? Amino acid activation, as I alluded to earlier, is when the amino acid gets attached to its specific transfer RNA. The cell has to know which transfer RNA to put which amino acid onto. And the way that the transfer RNAs are differing from each other is in their anti-codon. Right? So a cell that has an anti-codon of GCU, for example, is going to have a specific amino acid that'll be put onto its end. A different transfer RNA that has a sequence UUG, okay, is going to have a different amino acid attached to its end. All right? Now, the attachment of the proper amino acid to the end of the transfer RNA is absolutely essential. If you don't put the right one on, then the transfer RNA is going to carry the wrong amino acid to be put into the protein. So this activation step is pretty critical in making sure that the cell makes the protein in the right way. Okay? Well, there's a bunch of steps in the process. I'm just going to sort of talk you through it. We see the overall process occurring right up here. Amino, acyl, tRNA. Okay? So what does this mean? Here's a tRNA that doesn't have an amino acid onto it. Its end is empty. The end is empty. Okay? Here is an amino acid, and as we've seen already in class, things that make things happen in cells are enzymes that catalyze reactions, and the reaction that's getting catalyzed here is the linkage, that is the attachment covalently of the amino acid to the transfer RNA. So you can see that's happened over here. These guys have gotten attached. These enzymes that do this are pretty interesting. They have to look at the anti-codon and they have to attach the right thing up at the other end of the tRNA. They're kind of like a DNA polymerase, which remember had to read the other strand and then put the right base in complementary to it. This guy has to read the anti-codon and put the right base up at the other end of the transfer RNA. These enzymes have a name that you should know. They're called amino acyl tRNA synthetase. Mouthful of a name. And the most common question I get is there an abbreviation for that name? Well, the bad news is they're not, but I'm open to suggestions about what we might call it. Amino acyl tRNA synthetase. So I leave it up to my classes sometimes to decide on a class nickname for an enzyme. And I'm going to ask you the nickname for this enzyme that you will all remember. Ahern? We don't want to think in those terms. That's too bad. The what? Bob? Bob is simple. How many would like Bob? Or do you have another name to suggest? We can go for other names. What are other suggestions? ATS. You're getting very literal here, aren't you? Okay. That's good. That's good. You'd like Bob? Well, I'll be fair to everybody. Anybody else have other names to throw into the hat? You see, I love it. After you guys finish this class, you're going to go out, and the way you're going to remember this is, you're going to talk about this enzyme, and your boss is going to be talking, and you say, oh, that's Bob. Right? Okay, so we have ATS, we have Ahern, and we have Bob. And I'm going to kind of pull Ahern out. I think we don't want to. I shudder to think what people say about Ahern already, so that's probably not good. So how about Bob versus ATS? ATS? Bob? I think Bob's got it. Okay. So if I ask you about that on exam, Bob is a perfectly acceptable answer. Please don't take it outside this class. Okay? All right. So Bob catalyzes the covalent joining between the amino acid and the proper tRNA. That's pretty cool. Okay? Without Bob, we don't have proteins made the right way. Because then the wrong amino acid would be put onto the tRNAs, and the tRNA carries the wrong amino acid to the ribosome, the wrong protein is going to be made. We've got a real problem. So Bob is very important in that respect. Now, one other thing I'll tell you, we'll talk about this not today, but we'll talk about it next time, is that this bond between the amino acid and the tRNA is extremely unstable. It's extremely unstable. It's so unstable that if it doesn't get covered up by a protein very quickly, its half-life is about a second. It falls apart. This bond right here is very unstable. So once Bob puts these two guys together, there are proteins that come along that cover that up to prevent water from breaking the bond. It's actually water that does that. So this bond has to be protected from water. Okay. Now, I know you guys would probably like for me to go through all this, but I'm not going to do that. Sorry to break your hearts. Okay. Now, I showed you the structure of a tRNA schematically as a clover leaf, but in fact, that's only a two-dimensional representation. This guy is in a projection of 3 dimensions. What it actually looks like, it's kind of a bent-over structure. Okay? And you can see a whole bunch of things here. This is called the pseudo-uridine loop here. And the pseudo-uridine is one of the modifications chemically that's occurring in here. You don't need to know that. But down here, again, this is the anti-codon. This anti-codon is really critical for the identity of this tRNA. Okay? It's up here at the other end of this guy, the 3' end of the tRNA, where the amino acid will get attached. So the amino acid gets attached to the 3' end. If you're wondering where the 5' end is, it's right there. So if you trace it all the way around, blah, 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 you come up here, eventually you get to the 3' end. So the 3' end is where the amino acid gets attached. The anti-codon is what the enzyme, Bob, is looking at to put the right thing on there. Okay. Let's see. tRNA structure. Okay. So now we're going to split. We're going to talk about prokaryotes. And I know Mom's here and she doesn't want to stay the whole period, so we're going to make it short today. All right. I will. Yes. I hear that. Yes. But I do need to start prokaryotes. Okay. So let's start talking about prokaryotes. And then we'll have a thing from Mom at the end. All right. Prokaryotes. This is only in prokaryotes. Yeah. The process of translation, which I'm going to show you not today, but I'm going to show you next time, involves a step called initiation, a step called elongation, and a step called termination. All right? That elongation goes over and over and over, each time adding one more amino acid until the final protein gets made. All right? One of the things I lied about a little bit earlier in the lecture was I said all proteins, or essentially all proteins, start with methionine. And that's true, except in prokaryotes, the methionine is actually chemically modified. The very first methionine that gets put into a protein is chemically different than all of the other methionines, because yes, methionine can appear in other places in a protein. Okay? The very first one is modified chemically. It's called formyl-methionine. Right there. And that means that this group gets attached to the amino end of the very first methionine. Now, there's a chemical reason why that happens. I'm not going to go into it here, but suffice it to say that you do need to know that the very first one that's made is called formyl-methionine. We don't see that happen in eukaryotes, and this only happens in prokaryotes. Okay. In the process of initiation, which is what I'm going to start right here, okay, the overall complex has to be assembled. Now, this is a pretty cool process that's orchestrated inside of cells. The ribosome, which is what I described as the factory, exists in 2 giant subunits, total of about 70 proteins. There's one that's called the large subunit and one that's called the small subunit. You put them together and people sort of draw them like a snowman, where the large one is the bottom part of the snowman and the small one is the top part of the snowman. Okay? In prokaryotes, the small one is called the 30S subunit. The large one's called the 50S subunit. Okay? Now, all I'm going to show you of the initiation process today is the fact that in the assembly process, remember we've got to get a messenger RNA in here, and we've got to get things started. In the assembly process, it's very important that the ribosome know where do I start translating the sequence. The sequence that it translates doesn't start at the very end. It's actually in a few bases. And moreover, it doesn't always start with the first time it sees AUG, which is the first codon. So the question is, how does the ribosome know which AUG to start at? Okay? Well, the answer is that the 30S subunit has ribosomal RNA in it. This is an RNA molecule within the 30S subunit. And part of the sequence of that RNA is complementary to a sequence right next to the proper AUG. Now, what that means then is that during this assembly process, this sequence called the Shine-Dalgarno sequence, right there in the messenger RNA, forms a base pair with the ribosomal RNA sequence that puts the AUG in exactly the right place. So I'm going to repeat that. It's a little complicated. All right? The 30S subunit has a ribosomal RNA. That ribosomal RNA has a sequence that's complementary to a region of the messenger RNA. That region of the messenger RNA is called the Shine-Dalgarno sequence. We put these guys together. Yeah, it's right there. It's S-H-I-N-E. Oh, you can't see it from back there? Sorry about that. S-H-I-N-E dash D-A-L-G-A-R-N-O. And those were the names of the two people that discovered it. Okay? When that base pairing happens, it aligns the AUG in exactly the right place for translation to happen. Now, because it's a little deep, I probably will repeat that at the beginning of the next lecture. Yes, sir? It's not an intron because, again, we're talking about prokaryotic cells, so there are no introns in prokaryotic cells. So this is something that's found next to the AUG of almost every prokaryotic AUG. I hear everybody getting ready to go. Would you like to have a song to summarize what we've learned? Okay. It's very easy to sing this one. It's to the tune of America the Beautiful. Oh, beautiful with RNA to make the peptide bonds. You hold tRNA so it can pair up with codons. The ribosome, translate mRNA. Initiate and translocate from start to UGA. See you guys Monday.